Let us continue where we left off, the usage of nosodes in homeopathy. Hi friends, I am Dr. Saurabh Arora, and we are going to continue our discussion on the usage of nosodes in homeopathy. In our previous talk, we had an elaborate discussion on the usage of nosodes in homeopathy, which was based on the outcome of a survey. And we saw that the nosodes are used in the clinical practice without any hindrance or without any problem. But does that correlate with the research too? So in today's talk, we are going to discuss few of the peer-reviewed scientific publications in which nosodes were used and the outcomes were promising. We are going to have some translational values, some challenges, some key questions arising out of these studies. I have categorized these studies according to the years of publication. So the first study we have with us is a study by A. Chatterjee and the team, which was published in the year 2012 in the Journal of Alternative and Complementary Medicine. In this study, 3 cases of stage 4 metastasis, i.e. stage 4 of various carcinomas were treated with the Sorinum 6X. These 3 cases were of periampullary, gallbladder and liver metastasis. The Sorinum 6X was given to the patients in a predefined format for almost 2 years, and they were allowed to continue their existing therapies and treatment whatever they were taking. The study concluded that there was no adverse event related to Sorinum 6X in any form, and they maintained a stable quality of life in all 3 challenged cases. One may argue that case series are at the bottom of the pyramid of evidence, so why to give undue importance to such research in homeopathy? But I believe at least there is some evidence, because you can't see these kind of cases in everyday's practice because of ethical reasons, because of challenges, because of the expectation of the patients from homeopathy. But in this case, the homeopathy was used confidently in the 3 cases, and that to help the patient to a certain extent. The research design might be a challenge to us, but the outcome should be the motivation to research further into this area and to draw further research design based on the outcomes of this study. The next study we have was published in the year 2014 in the Journal of Complementary and Integrative Medicine by Upma Bagai and team. In this study, the anti-malarial efficacy of a combination of drug was used. So they took 2 drugs, one is chalidonium and other is malaria nosode. Both of them were taken in the 30 CH potency, and they were evaluated in vivo against the P. burgei infection. The hepatic and the renal function and the biomarkers were assessed, and the combination demonstrated the considerable in vivo anti-malarial activity with chemosuppression. The combination also enhanced the mean survival time of the mice. The liver and the kidney biomarkers were significantly altered in the Verum group than in the placebo group. So the study proposed that there is an effectiveness of this combination of chalidonium and malaria nosode being an anti-malarial agent against the P. burgei infection, and also safeguarding the hepatic and renal toxicity of the mice. So one may argue here that why a combination? Why not a single remedy? If you can recall the talk where we discuss about the difference between the ultra high dilutions and homeopathy, in this study what I believe, they studied the effect of the ultra highly diluted substances against the malaria. And there is a possibility, because since this team has been involved in this kind of research for a long period of time, it's a matter of curiosity. It's a matter of research question whether these agents are working or not. So the future research, one may have 3 separate groups of a chalidonium and malaria nosode along with the placebo. Moving further, we have a study from a very renowned author in this field, a renowned scientist, Professor A.R. Khudabakhsh, who is an authority in the biomedical research. Professor A.R. Khudabakhsh published this study in the year 2016 in the Journal of Integrative Medicine. And this study was to assess the action of the Sorinum 6X on the lung cancer cell lines. So they took 3 cancer cell lines, A549, MCF7 and HepG2, and Sorinum 6X along with the ethanol 6X as a control was applied on these cancer cell lines. The treatment effects were studied on the cell cycle arrest, cell death, and generation of the reactive oxygen species, and changes in the mitochondrial membrane potential, which were assessed using flow cytometry and fluorescence microscopy. The expression of several signal proteins related to the apoptosis and cell survival was quantified using the Western blotting method and the confocal microscopy. The treatment of the cancer cell lines with Sorinum showed showed greater anti-cancer activity in the A549 cell line, where the proliferation of the cancer cells were arrested 24 hours after the treatment. So it was concluded that the Sorinum 6X triggered apoptosis in the A549 cell line via both up and down regulation of the relevant signal proteins, including p53, caspase-3, and BAX and BCL2. Now, this study was a prominent study because when you're researching into the cancer cell lines, it is very difficult to have the outcomes, and Sorinum 6X was able to induce those outcomes. Moving further, we have a study where the effect of the homeopathic medicine and nosode prepared from the C. homnivorax was used. This C. homnivorax is the major fly-causing myces in the livestock. In the conventional treatment, chemosynthetic insecticides are used, but they come with a challenge. So to study the efficacy of homeopathy in myces induced by the C. homnivorax, this study was undertaken. It evaluated the use of homeopathic medicine sulfur and pyrogenum, and also the nosode prepared from the same material, the C. homnivorax. And they were used in the 8CH and 12CH potencies against the third stage of the larvae of the C. homnivorax. They found that the mortality rate of the larvae were 94.6% in the sulfur, but it was 98.6% in the pyrogenum group. The overall results suggested that the homeopathy could be used therapeutically to prevent and to treat animals and humans with the myces caused by the C. homnivorax. This study is again an important study because when it comes to infections where there are limitations in the conventional medicines, we need to have the alternative systems which may offer a safe and efficacious mode of treatment. Moving further, we have a study published by P.A.A. Raj et al. In the year 2020. And this was to evaluate the ameliorative potential of the homeopathic drugs in combination of sulfur, thuja, graphitis, and Sorinum 30C in 16 dogs which were affected with the oral papillomatosis, and they were not treated at all before with any kind of treatment. The homeopathic combination of the drug was used in comparison to the placebo, and it was given twice daily to these dogs for 15 days. The homeopathic treatment group showed an early recovery and a significant reduction in the lesions in comparison to the placebo-controlled groups. The study proposed that the combination of homeopathy drugs help in fastening the regression of the oral papillomatosis, and it proves to be a safe and efficacious and cost-effective mode of treatment. One may ask why combination of drugs. In veterinary and in agro, where the usage of high dilutions are not strictly based upon the direct principles because these cannot be elicited, the combination of drugs are being used. Next, we have a study on the effect of the nosode on the lettuce, which was parasitized by the M. enterolobi. This study was published by Ferreira T.M. et al. In the journal Homeopathy in the year 2021, and the study investigated the effects of the M. enterolobi nosode resistance on the lettuce plant. The nosode in various potency was applied to the lettuce plants through irrigation before the seedling has set in. And once the seedling has set in, it was subjected to the nematode inoculation. The M. enterolobi nosode significantly reduced the nematode production in the plants and also affected the root density, showing that this nosode can play a pivotal role when it comes to the infection, and it can be safely used to tackle these kind of infections to improve the yield and to reduce the disease burden in the plants. Moving further, we have a study by Suri M. et al. Which was published in the journal Homeopathy in the year 2022, where the anti-malarial efficacy of the malaria nosode was assessed. The nosode were prepared from the 3 strains of the P. falciparum. The in vitro anti-malarial activity was assessed in the schizont inhibition assay. The mice were inoculated with the P. burgii infections, and the treatment was initiated on the same day. They did the biochemical, cytokine, and histopathological markers as a standard method. The mixture nosode, which was prepared from 3 strains, exhibited promising anti-malarial activity against P. falciparum and P. burgii infections in mice. And the histopathological and the biochemical assay showed that these remedies were safe to liver and kidney. Coming to the end part of the talk, these are the few studies we have researched upon today, and these studies are showing the promising results of the nosodes. But the story doesn't end here. All these studies, whether they are flawed, whether they are strong, whether they are robust, actually they are contributing to the research aspects in homeopathy. We should be having research question out of the study for the future research. Research is not always having the positive outcomes, but research is all about having the results which are reliable and which can be replicated. So with this, we'll come again with one more question with few more studies and try to have the translational values for the same. Till then, stay tuned, because homeopathy works.